Welcome to this symposium. It's with great pleasure that I can introduce to you Professor Gerard Muyzer, who is here in Mallorca from the University of Amsterdam in the Netherlands, and who will give a talk about microbiome in seagrass. So very interesting. Thank you for that.
Okay. Buenos dias. Thank you, Iris, for these announcements. And welcome to my symposium, to my presentation. And so what you see here is not the lawn of the Johan Cruyff football stadium in Amsterdam. It's actually the seagrass meadows of the coast of Mallorca. And these plants, they actually live in intimate relationship with bacteria. And that is actually the topic I will discuss with you. So the seagrass microbiome. And seagrasses are marine flowering plants, and they actually form dense meadows in coastal areas. And the flowering plants is that you see here, the very nice fruits of Posidonia. And actually they form dense meadows in coastal areas. They're highly productive ecosystems. They're ecosystem engineers because they stabilize sediments and they actually take out particles, and they are involved in nutrient cycling. And also very important, they are a nursery and feeding habitat for many animals. And as I mentioned before, I mean, these marine plants, they're living in intimate relationship with microbes. And so there are microbes on the leaves, which will I not discuss. And there are also microbes in the rhizosphere of the seagrasses. And I will call that the rhizobiome. So the rhizosphere in its microbiome is the rhizobiome. And this is a cartoon from people who work on terrestrial plants. And you see they have, let's say, bacteria which are the good. They actually, for instance, they provide a tolerance to abiotic stress, or they do nutrient acquisition, or they protect against pathogens. And there is also, let's say, organisms, microorganisms that are the bad. They actually cause plant diseases. And then there is also the ugly, for instance, for spoiling food. And so if you look at the biogeochemical processes in the rhizosphere, then actually we see, and here you see the rhizosphere for one of these seagrasses. Then you see here actually the root. This is the root. And then at daytime, of course, there is a lot of oxygen produced by the plant, and that's also sent to the root system. And so you see that there is, let's say, a large production of oxygen. And because, let's say there is a lot of sulfide in the seawater, then sulfide reducers might produce sulfide. And sulfide is a toxic. It's a toxic for us because it actually binds to the iron in our hemoglobin. But it is also a toxin for plants, for seagrasses. Yeah. And what you see at night, then of course there is no oxygen produced, and then you see that the production of sulfide is a lot, and so therefore they might also influence, of course, the seagrasses. So the oxic rhizosphere and the anoxic rhizosphere with different, let's say, bacterial groups. And so the aim and the research questions are the following. We want to obtain, of course, insight into the interactions between the seagrasses and their microbiome. And then we have some specific research questions, and that is, for instance, is the rhizobiome plant specific? Does the rhizobiome co-evolve with its host? And this is more or less the idea behind the holobiont theory. Who are the keystone species in the rhizobiome? And of course, what role do these species play? And I hope to give answers to these questions in the presentation. And so what we did is actually we collected samples from the rhizosphere of 3 seagrasses, Zostera marina, Zostera noltii, and Cimodocea nodosa from 2 sites, from Portugal and from France. We extracted the DNA to do 16 Amplicon sequencing as well as metagenomic sequencing. And we did some incubation experiments of the plants at different sulfide concentrations. And after that, we used fish, I will explain that later on, and confocal laser scanning microscopy. We did microelectron measurements, and we measured the, let's say, the photosynthesis and the growth of the plants. So first, because I don't know your background and you might not be familiar with all these, let's say, novel methods, just a brief explanation of the principle of, let's say, 16S Amplicon sequencing. And what you see here are, for instance, let's say you extract the DNA and then you amplify the 16S ribosome RNA gene with primers that have a different barcode. And that is illustrated by the different colors here. And so you amplify, for instance, you have the 3 plants or you have replicates. I will come to that in a moment of a plant. And then you amplify the 16S ribosome RNA gene, and then you actually mix it. You pull the amplicons, and then you sequence it with one of these, let's say, next generation sequencing devices. And in this case, it's the Illumina Muyzer. And then when you get the data, you actually use, let's say, a software program, in this case, QIIME, and that is the gold standard for amplicon sequencing. And then what you see actually here is that you get the different reads with the different barcodes. And so you know exactly that the rare barcode comes from this DNA, for instance. And then by the software, you can do all kinds, let's say, analysis to get your results and to identify the microorganisms which are present in the rhizobiome. And so let's look at the first results. And what Katarina Kutsho did at my PhD student is she actually did the Amplicon sequencing. She did the Amplicon sequencing on 5 replicates. And that is very important because that gives you some statistical power. Yeah. And you see here, let's say the bar graphs of Zostera marina, noltii, Cimodocea, the sediment and the seawater as references. And actually, if you look at this bar graph, it's very difficult to see the difference. But as we then use, for instance, principal component analysis, you actually see the following results. And so here in blue, the blue squares are the microbiome of the seawater. So the bacteria which are present in the seawater. The yellow triangles are the microbiome of the bulk sediment. Yeah, so outside the rhizosphere. And then here you see, let's say, the microbial communities which are present in the rhizosphere. So the rhizobiome of the 3 different seagrasses, marina, noltii, and Cimodocea. And what you see excellently is that the seawater and the sediment are actually separated from the microbial communities in the rhizosphere. And from this we can actually mention that the rhizobiome or the microbiome is plant specific. It's different from the bacteria which are present in the environmental samples. If we then come to the next question, so does the rhizobiome co-evolve with its host? Then the answer is no, because what you see here is the different seagrasses, so Zostera marina from Portugal, Zostera noltii from Portugal, Zostera marina from France, and Zostera noltii from France. And actually what you see is that the seagrasses, marina and noltii from Portugal, they actually cluster together, and they are separated from the rhizobiomes of the seagrasses, which are collected from France. And so this indicates that there is no, let's say, co-evolvement of the host and its microbiome. Because in that case, you would actually see that Zostera marina from Portugal and Zostera marina from France would be actually grouping together. And Zostera noltii from Portugal and Zostera noltii from France would group together. And that is not what you see. So the rhizobiome does not co-evolve with its host. If you then look at the keystone species, then we make a kind of, let's say, a core community. So we compare all the, let's say, the OTUs of the different samples, Solstera marina from Portugal, Cimidocea, Solstera marina from France, Nolty from France, Nolty from Portugal, and that you see that the core community consists of 101 OTUs, and the most of these OTUs are, let's say, affiliated to the proteobacteria. And I can tell you it's not on this slide, but it's mostly the gamma proteobacteria and the delta proteobacteria. And so these are more or less the keystone species, but in particular, let's say, the proteobacteria. So if you then look at niche differentiation of, in this case, sulfate-reducing bacteria, which belong to the delta proteobacteria, then we see, here is the, let's say, the proportion of sequences. Here we see the sediment, so the sulfate-reducing bacteria, or in this case, the Desulfobacteriaceae, which is a family of the sulfate, of the delta proteobacteria. This is the result of the sediment, the bulk sediment of France, the bulk sediment of Portugal. Here you see the seagrasses from France, the seagrasses from Portugal, and again, the water, the seawater as a let's say, as a reference. And then you see that, especially in the seagrasses, in the rhizobiome of Portugal, you see a very high number of sequences belonging to the Desulfobacteriaceae. Here we see another family within the delta proteobacteria, the Desulfogobaceae, and what you see here, you see a little bit more in the sediment, which is logical because there are also delta or sulfate-inducing bacteria in the sediment. Yet we don't see any in the seawater because you don't expect them to be in the seawater because they are anaerobe, and so they're not in the aerobic seawater. And you see that they are, let's say, both present, or they're present in both, let's say, the seagrasses of France and the seagrasses of Portugal. So you see that that especially for the Desulfobacteriaceae, they're mostly present in the seagrasses of Portugal and not in the seagrasses in France. If we now look at the sulfur-oxidizing bacteria, then we see actually a difference. So we have, let's say, sulfur-oxidizing bacteria, which belong to the gamma proteobacteria. For instance, the Chromatiaceae and the Trichiaceae. Yeah. And we have sulfur-oxidizing bacteria, which belong to the epsilon proteobacteria, like the Campylobacteriaceae and the Helicobacteriaceae. And also here you see the sediment, the seagrasses from France and Portugal, the seawater, and you see that the Chromatiaceae are mostly present, let's say, in the seagrasses of Portugal. What you see here is that epsilon proteobacteria are mostly present in the seagrasses of France. The same as here, you see that another group of sulfur-oxidizing bacteria, they are present in Portugal and a little bit in France. And here you see the opposite. So what you can say is that in France, it's mostly the rhizobiome, or at least the sulfur-oxidizing bacteria, are mostly belonging to epsilon proteobacteria, while in Portugal, you will see that the sulfur-oxidizing bacteria are mostly, let's say, present from the gamma proteobacteria. So, as I mentioned, the number of replicates gives you statistical power, and that means that you can also do, let's say, this kind of co-occurrence network of the rhizobiome. Yeah. And you see, don't look in detail because this is just an overview. And you see that here there are different, let's say, taxa present, and you see green lines and red lines. And so the green lines is a positive interaction. And so you can think at stimulation or facilitation. And the red lines are actually negative correlations. Yeah, and there you can think of competition. And this is, of course, too difficult to get something out of it. And so if we only look at the sulfur bacteria, then of course we see a very nice network, a very nice small network. And we see here the different let's say, sulfur-oxidising bacteria, which we have seen before, and we see here the sulfate-reducing bacteria. And you see the sulfate-reducing bacteria, they actually convert sulfate into sulfide, which is then used by the sulfur-oxidising bacteria to convert into either elemental sulfur or sulfate. And what you see is there are green lines, let's say positive interactions between the sulfate-reducing bacteria and these sulfur-oxidising bacteria. And there are red lines, and so actually the sulfide, which is, let's say, produced by the sulfate-reducing bacteria, might stimulate, let's say, the sulfur-oxidising bacteria. And you might see red lines between the different sulfur-oxidising bacteria, and that might point to a kind of competition. So coming to the, let's say, the summary of this 16 Amplicon results. So the microbial communities are plant-specific, as already mentioned. The rhizobiomes do not co-evolve with their host. Sulfobacteria are keystone species, so both, let's say, sulfate-reducing bacteria as sulfur-oxidising bacteria. And sulfur-oxidising bacteria show a niche differentiation. So now to the next part which I want to discuss, and that is the sulfide incubation experiment. And for this, we actually planted the seagrass shoots into agar with sulfide. Yeah, so it's a, let's say, different concentration of sulfide, sodium sulfide. And of course, that might diffuse out and might have an influence on the seagrass itself, and of course the rhizobiome. And then we use microelectrodes for, and here you see, so this is a drawing, but here you see a photograph, and here you see one of these microelectrodes with a micromanipulator, and here you see it also, and you see it has a very small tip. Yeah, and with the micromanipulator, you can go into the agarose. I had hoped that it would not break anything because the agarose is soft, but actually the PhD student and the master student broke a lot. And actually we measured inside the rhizosphere, you see here, and we measured outside the rhizosphere. Yeah. And let's look at the results. And so here are the graphs. This is the depth into the agarose. And this is, for instance, here with the pH. So we measured pH, we measured oxygen, and we measured sulfide. And this is the oxygen or the pH. And you see that the red line is the pH outside the rhizosphere, and the blue line is the pH inside the rhizosphere. And you see that the pH inside the rhizosphere is lower than the pH outside the rhizosphere. Why that is, we don't know, but you can imagine that if sulfide is oxidized completely to sulfate, that protons are released, which might actually decrease the pH. Yeah. If you now look at the oxygen, and that we measured on let's say 4 different days, day 3, 6, 9, and 10, and then you see that the oxygen is disappearing, let's say, very quickly. Probably because it's taken up by the heterotrophic bacteria. If you then look at the sulfide, and again the sulfide inside the rhizosphere and the sulfide outside the rhizosphere, measured on 4 different days, and you see actually that the sulfide, let's say, out or the sulfide inside the rhizosphere is lower than the sulfide outside the rhizosphere, which might indicate that something is taking away the sulfide, yeah, is neutralizing or detoxifying the sulfide. So as I mentioned here, we use FISH, and that stands for fluorescence in situ hybridization. And this shows the principle. If you have a sample, in our case, let's say part of the rhizosphere, you fix it with paraformaldehyde, and then the cells are not only fixed, but they're also permeabilized. Then you incubate them with, let's say, oligonucleotide probes with different fluorochromes. and they might actually bind to the target RNA in your cells. You wash it, and then actually you visualize it under the epifluorescent microscope, or in our case, the confocal microscope. So this is just, and you see, this is a kind of optical section in the 3D image of your of the root system with the different bacteria on it. And you see, if you actually go through with this confocal laser microscope, go through your specimen, then actually you see different layers in the root system. You might see the fluorescent staining. You see, and this is just an illustration to give you an idea. However, if we then look at the results before sulfide incubation, and this is, let's say, a probe which is made for the gamma proteobacteria, and you know that the gamma proteobacteria that have these sulfur-oxidising bacteria, you see that, and here there's a probe for the delta proteobacteria, and those are the sulfate-reducing bacteria. And so actually you see different colors because I think that the general probe for bacteria was in red. The specific probe for the gamma proteobacteria and for the delta proteobacteria was in green, and the blue staining was the DAPI staining, which actually stains the general DNA. And so you see that there are some of the cells are actually yellow because, of course, they are stained by the red probe and the green probe, and so they're yellow or orange. You also see it here. But actually, if you do the same fish analysis after sulfide incubation, then, and this is an enlargement of this part, and again, this is an enlargement of this part, then you might see that there are more, let's say, gamma proteobacteria and delta proteobacteria present on the root system. Looking at the sulfide on the photosynthesis and growth, and so we looked at the photosynthetic activity over different days at two different sulfide concentrations, 10 micromolar and 500 micromolar. And actually what you see is actually at the end, at day 12, you see actually a statistic significant decrease, slight decrease of the photosynthetic activity. Yeah. Of course, if we would maybe incubate it for a longer time, or if we maybe add more sulfide, then of course the effect might be more visible. So little, let's say, influence on the photosynthetic activity, but more influence on the growth. And that is what you see here. We could only, let's say, measure the growth in Zostera marina because Zostera nolti is too small. The leaves are actually too small. And what you see here, the different sulfide concentrations, 10 micromolar and 500 micromolar. You see that at 500 micromolar, there are less leaves present. The leaf length is shorter. The new leaf length is shorter. The new leaf area is less, and the highest growth is also less. So you see, especially in the measurement of the growth of the seagrasses, you see a more, let's say, a dramatic effect of the high concentration of sulfide. So a summary of the sulfide experiment. Sulfide concentration in the rhizosphere was lower than outside the rhizosphere. Sulfide had a minor influence on the photosynthesis. However, growth was affected. The number of gamma and delta proteobacteria increased after incubation with sulfide. And the sulfide concentration in the rhizosphere might be regulated by oxygen from the roots, as I've shown you before. The plant is pumping oxygen, and that might actually bind chemically to the sulfide. It might also be the oxidation of the sulfur-oxidising, sulfide-oxidising bacteria, as I mentioned. That might also, let's say, lower the pH, or there might be a kind of precipitation with iron. If there's a lot of iron present and there is sulfide, then of course the iron and the sulfide will bind to the iron. And that points more or less to the black, let's say, the black color which we saw in the middle of the rhizosphere. Yeah. Of course, yeah, these are the different options, and probably you have to do more work to find out which of them is the most important one. So coming to the last part of my lecture, and that is metagenomics. And also again, for those who are not familiar with metagenomics, just a brief, let's say, explanation. So you extract the DNA, you fragment it, and then you sequence it completely. So what you see is the difference between the 16S, let's say, Amplicon sequencing and metagenomics is that with Amplicon sequencing, you do a PCR step, which might bias. And with metagenomic sequencing, you actually just extract all the DNA and you sequence it completely. And so what you get is many sequences. And then, of course, you need powerful computers to assemble these short sequence reads, which are about 150 nucleotides, into, let's say, contigs, into longer sequence fragments. Then actually you look for the, let's say, the gene finding and annotation. And then what you do is you have still a mixture. You do what they call phylogenetic binding, or binning, yes. And you actually see, okay, this belongs to this, let's say, bin. This one belongs to that bin. These, let's say, contexts belong to this bin. And so then afterwards you check the, let's say, contamination there, because you can actually put maybe one red contact into a green bin. And of course, that is not what you want. And so you can check the contamination with the software program. And if you then, let's say, are satisfied and you have nearly complete genomes of these individual, let's say, microorganisms, and we call that MAGs, metagenome-assembled-genomes, then actually you can do metabolic reconstruction. And so if you look back at the 16 Amplicon sequencing, there we could only identify who is there, but we cannot say about the function of these microorganisms. But with metagenomics, you can actually get, you can restruct the metabolic pathways, and you can then actually see what they might do. And so if you look at the taxonomic classification, you can also identify, of course, these MAGs, but this is then based on sequence reads. Then again, here we see Zostera marina from Portugal. Here we see Zostera marina from France. And again, we see that most of the sequence reads are together, let's say, clustering with the delta proteobacteria and the gamma proteobacteria. So it actually confirms what we found with the 16S Amplicon sequencing. Okay, if we then do a phylogenetic tree, and this is let's say, phylogenetic tree not based on a single gene, not based on the 16S, but based on, let's say, on complete genomes. And then we see here these, let's say, in black. I don't know if it's visible, but here we see, let's say, the MAGs which we found and which were from a very high quality, so a large completion and no contamination. And you actually see that they cluster at least these three, the CO1, the CO2, and the PT3F. They cluster with, let's say, genomes from symbionts from bivalves. Yeah, Solomia valum. Yeah, and also this one, I mean, it's a little bit further away, but at least these one actually they cluster. They're, let's say, yeah, they cluster together with genomes of this symbiont. So if we then look at the conceptual model of metabolic pathways, as I mentioned, and now it is possible, then again, at daytime, when there is a low sulfide and a lot of oxygen, we might see that in the aerobic part of the sediment, we actually found that this one of these MAGs, they had let's say, genes for different CO2 fixing pathways. So, and there are also genes for, let's say, different genes for sulfide oxidation pathways. So they had FCC and XQR for oxidizing sulfide, and they had the Kelvin cycle and the TCA, the reverse TCA cycle for CO2 fixation. And so what you see is that when there's a lot of oxygen and low sulfide, you see that they actually use, in the aerobic part, they use a different pathway for sulfide oxidation. And if it's, let's say, anaerobic or microaerobic, they actually use a different pathway for carbon fixation. Yeah. Furthermore, you see that here they actually perform denitrification, so actually you have from nitrate to nitrogen gas, and especially in this part when there's high sulfide at night, then they actually do dissimilatory nitrate reduction to ammonium, and that actually is one of the preferred, let's say, nitrogen sources for the seagrasses. And you see that all these names here, they refer to genes which we actually found in the metabolic pathways of this one organism. So coming to then I mean, as I mentioned, so sulfide oxidizing endosimians in mollusks, and this is the work of Chisse van der Heijden. And actually, what he found is that these seagrasses, they have a lot of, let's say, bivalves, and the bivalves have sulfide oxidation gill bacteria. And so actually when there is organic matter from the seagrasses, then the sulfide-reducing bacteria produce a lot of sulfide, and then actually the bivalves or the sulfide oxidizing endosimians in the bivalves, they actually detoxify the sulfide for the seagrasses. Now, in Portugal and in France, we looked for these bivalves, but we actually could not find them. So in our case, I mean, it was not the bivalves; it were really the, let's say, the free living symbionts in the sediment. And so, coming to the summary of the metagenomics. So sulfur-oxidizing bacteria related to endosimians are dominantly present in the rhizobiome. The seagrass meadows might be a source of free-living endosimians for mollusks. You can imagine that maybe if mollusks come, then they might take up these free-living endosimians. So in at least one of these are found various genes involved in the carbon, sulfur, and nitrogen cycling, which indicate a great metabolic flexibility. And so these sulfide oxidizing bacteria, they contribute to the detoxification of sulfide, and so we can actually classify as they belong to the good. And this is my acknowledgement. So Katarina, who is the PhD student working on the project, Tim, who is actually the master student, actually after this they got married and they have now 2 children. And then Ashvi and Egon, who is from Sicimar in Faro in Portugal, and all work was, or let's say the field work was all done in Portugal. And then we have Lex, Erik, and Joao, who also contributed to it. And so this is my talk, and thank you very much for your attention. And if you have questions, then I will welcome to answer those.
So there is a 35-second delay between the mission of your talk. So there's no questions yet in the YouTube channel. So maybe we can start with questions from the room.
Fine, yes.
Yeah, I have some questions. Thank you, Gerard, for this nice talk. And I have many questions, but I will only have some for you now. So
we can always discuss it afterwards.
Yes, of course. So one, you are looking to different locations and you see the rhizosphere, so the microbiomes are different from the same plant. Have you thought on looking at the endosphere of these organisms? Because our experience with other plants is they look much more alike when the endosphere and the exosphere.
Yeah, no, we have not looked at the endosphere. No, so that could be that these, let's say, the endospheres are more similar.
Yeah, because they select the organism somehow from the rhizosphere and they go into the endosphere.
Yeah, I mean there is already some work done here on the characterization of the microbiomes of seagrasses, the leaves, the seagrass, the phylosphere, the rhizosphere, and the endosphere. But it's very difficult, let's say, to compare these results because everyone used different, let's say, different seagrasses, different seagrass species, and they actually use, let's say, different methods to collect the samples.
I have another one, if nobody. So, do you have with the sulfur-reducing organisms that you found, do you see any relation between being complete oxidizers or incomplete oxidizers? Do you see a relation of who is there? And
Actually, I showed you the results at the high taxonomic level. I think that if we go maybe deeper into the taxonomic level, then we might certainly see the difference.
So we need a little bit more fine-tuning probably to see this, huh?
Yes. But we can certainly we have the data, so we can certainly look into it.
I had more questions, but do you have any questions?
I have a question. Maybe you said so at the beginning of your talk, and I didn't pick up on it. But the seagrasses you looked at, did you look all at subtidal or intertidal? Where they were living, where they came from?
Yeah, I know. Seagrasses and Zostera marina, they're subtidal, and Zostera nolti is, of course, in the tidal area, which becomes, let's say, dry at low tide and covered with water at high tide. So yes, there is a difference, of course, between these.
And do you expect it to contribute a lot to differences in the microbiome?
What you actually saw in one of the slides is that, I don't know, I can go back, is that here is that Zostera, here, that Zostera marina and Zostera nolti are actually more or less clustering together. And that is this for Portugal, but it's the same for France. So yes, I mean, also here, if you maybe really look into great detail, you might see it. You could also say, okay, maybe if we, then you see that there are 3 of marina here, and there are 3 of marina here. But I think that marina and nolti, or the microbiome, the rhizobiome for marina and nolti, are very similar. And you see that in Portugal, and you see that in France.
Okay. So at the moment there are no more questions in the chat, so maybe you don't if you
I can make the last one, and this is more or less about the role of the sulfur-oxidizing organisms there. So they are autotrophic. So, and this is theoretically I was thinking more in a heterotrophic system, because the plant probably exudates also substrates for the organism living there. So what kind of role will they play in this environment? And if the numbers are big enough, or just is a question of a chance that they are there. So they are there, but they are not really.
No. I mean, if you look at the percentages of sequence reads, then you see that they're in large numbers, relatively large numbers. Okay.
What is relatively large?
I don't have to look into detail, but a few percent. Yeah. So the different the Chromathiaceae and the Campylobacteriaceae, they're in percentages. So it's not that they're in, let's say, 0.1%. Yeah. So they're really in percentages present. Yeah. Coming back to your first statement, that you would expect that they would be heterotrophic. Don't forget that there are many heterotrophic bacteria living in the rhizosphere, which might actually compete for all these cookies which are produced by the plants, compete with the let's say, sulfur-oxidizing bacteria. And so therefore it might, for the sulfur-oxidizing bacteria, be much better to be autotrophic, because then actually they can consume the CO2.
I was thinking more in a role. So because you said that probably could detoxify.
Yeah.
Help to detoxify. And my question was whether the numbers were high enough to really help in detoxifying the sulfate that is there.
They are high, but I mean, I also don't know if that is the only contribution to, let's say, detoxifying. You might also see that there might be a part might be chemically oxidation because of the oxygen produced by the plant. There might be part of, let's say, binding to iron, you get iron sulfide. But I mean, the number of, let's say, the percentage of sequence read belonging to sulfur-oxidizing bacteria is high.
Yeah. So in my last question regarding the pH decrease, Could not be also due to fermentation processes,
Yeah,
because fermentation will decrease also the pH.
yeah, that could be. Yeah, I could not exclude it. I mean, but it might also be, you know, from Begitoa, that actually, if it's let's say oxidizing sulfide to sulfur or sulfate, or especially to sulfate, that pH is decreasing. You see actually a decreasing pH in the rhizosphere, which you don't see outside the rhizosphere.
Yeah, perhaps it's a combination of both.
Yeah. Yeah.
Nice, very nice system.
Thank you. No further questions?
I know you have a last question about the microsensor work you did. Did you use light conditions only, right? Or did you also use dark conditions for the microsensor measurement?
No, I don't think that we use dark conditions. No.
So do you think the light intensity or dark conditions could change and even lead to a lower pH in the rhizosphere?
Yes, I mean, yes, that could be. I mean, you can imagine that fermentation might actually increase in the dark much better. Yeah. So that might be.
Yes.
Yeah.
Interesting. Last question.
There's no questions in the YouTube channel? No.
Oh, okay.
But we had a lot of questions in the room.
Okay.
If the people online have questions, they can also always contact me. Yeah. So.
Yes. So thank you very much for this
You're
really
welcome.
interesting talk.
Thank you very much for being here, and also the people online, if they're still there. Thank you very much.
Yes.
Okay. Thank you.
So I mean,
Oh wait, there's a last question.
Oh,
Did you already turn it off, or?
no.
So the question is, are there predictions of how global change will affect the spatial temporal dynamics of different functional groups of sulfur bacteria marine ecosystems?
Yeah. Yes. Yes. I didn't touch upon that, but let's say